Good afternoon to all. We will discuss another next topic. We will discuss about carbohydrate, protein, lipids. The next topic we reach on enzymes. That is one of the most important topic in biochemistry. We will discuss about enzymes, their general properties, their classification, their mechanism of action. Now some scientific equation like Michel-Menten and Landweber, mathematical tools to study their mechanism of action. And in the last we will study about allosteric enzymes. In the last we will study about allosteric enzymes. So first of all, we should know what are enzymes. Enzymes play important role in the metabolism, diagnosis, therapeutics. We know that all the biochemical items are basically digested by the enzymes. So enzymes play important part in the metabolism, in the diagnosis also, in the therapeutics also, to diagnose some disease and use in the treatment, or all some are based on enzymes activity. Like ELISA test, PCR test, they all are based on enzymes. In our body, in human beings, in fact in all living beings, all biochemical reactions are catalyzed by enzymes. Okay, all biochemical reactions are basically catalyzed by enzymes in the living organisms. However, the level of enzyme in blood is of diagnostic type. When we detect any enzymes from the blood, they inform us. It means they are a good indicator in disease like myocardial infection. In fact, they are good indicator in disease such as myocardial infection. Okay. They can be used therapeutically as digestive enzymes. Okay, they can be therapeutically used such as digestive enzymes. So these are the main very prime importance of enzymes. They can be used therapeutically as digestive enzymes. Okay, so what I told, enzymes are important role both in metabolism, diagnosis. In the blood, they can inform certain infections. They can carry out various reactions in the living body of human living organisms, and they are used also as in the digestion of various products. Basically, biochemically, all enzymes belong to proteins. Enzymes are proteins that increase the rate of reaction. Whenever there is certain digestion of the reaction, all reaction based on biochemical method. So enzymes are proteins that increase the rate of reaction by lowering the activation energy. Okay, this concept is little bit from the chemistry point of view, so we will not discuss in detail. We just know that enzymes are proteins which increase the rate of reaction by lowering down the energy of activation. They catalyze nearly all the chemical reactions. They catalyze nearly all the chemical reactions that take place inside the body. Okay, they catalyze nearly all the chemical reactions that takes place inside the cells of body. They are not altered or consumed during reaction. They are not altered or consumed during reaction. And they can be reusable. They can be reusable. So these are the main importance property of enzymes, that they belong to protein. They increase the rate of reaction by reducing the level of activation energy. They catalyze all kind of chemical reactions. They are not consumed. They remain in the same quantity, and they can be used again and again for different reactions. So these are the specific property of enzymes. Now there are some important terms to understand the biochemical nature of enzymes. First is active site. Active site is the area on the enzymes where the substrate or substrates attached to it is called active site. Whenever enzymes are used to perform any reaction, they cannot perform any reaction without the active site, because there is some surface on the enzyme, so some molecule which is called a substrate attached on the surface of the enzyme. So as a result, what will happen? Enzymes become activated. An active site just occupies only the small part of enzymes. As we know, enzymes are usually very large proteins, so the active site is just the small region of the enzyme molecule. Okay, the enzymes are usually very large proteins, and the active site is usually the small region of the enzyme molecule. Okay, means enzymes themselves, they are very large, but the site, the surface which is using by touching any other substrate, that is very, very small. We can see that figure. Uh, Enzyme molecules contain special pocket or cleft. This opening is called as cleft, where the active site, where it is called as active sites. This pocket-like structure are called as active sites, and see this diagram. Green color is the substrate. It is entering into the active site of the enzyme. After that, what will happen? Once the substrate is attached to the active site of the enzyme changes shape slightly. When shape, after going all reactions, finally, this enzyme will yield some products. So this is called as enzyme substrate complex. It is called enzyme products complex. It is finally products again leaving the site of enzyme. So very simple method is there. So what is this method? Substrate brings us substrate reaching on the active site of the enzyme. As a result, enzyme substrate complex is there. So enzyme will change their shape and size slightly. They will form enzyme products complex. This color from green is turned to be blue and red. Substrate is already blue and product is red. And finally, product is differentiated from the substrates. The products are leaving the active site on the enzyme. This is the main mechanism how the active sites come on the site of enzyme. Then we discuss about substrate. What is substrate? Any reactant in biochemical reaction is called as substrate. Any reactant in biochemical reaction is called a substrate. Means any molecules which will react to give certain products. So initially it is called as reactant. So the reactant in biochemical reaction is called as substrate. So when the substrate binds to an enzyme, it forms enzyme substrate complex. So whenever substrate binds to the enzyme, what they will form? Enzyme substrate complex. So in enzymatic reactions, the substances at the beginning of the process on which enzyme begins is called as substrate. The substance at the beginning of the process on which enzyme begins its action. So what enzyme will do? Enzyme will take action on the substrate, because it is the first compound which is coming towards the enzyme. So this compound on which enzyme is taking certain action is called as substrate. Okay, so it will call as what? It will call as substrate. You can easily see this figure. Then another method of enzyme action is lock and key method. Whenever we go outside our home, we put locks on the doors and we have the keys of the locks. When we return back, what we will do? Put the keys in the lock and the lock is open. Same concept apply here. This model is called as lock and key model. So in the lock and key model of enzyme, the active site has the rigid shape. The active site of the enzyme, that is called as lock, it is rigid shaped. So only substrate which are matching with the shape of this active site, only they will fit as a key is fit inside the lock. So here the active site is the lock and the substrate is the key. So exactly the shape of the substrate should match with the active site of the enzyme, so that can easily penetrate the active site, and thus the enzyme can perform the reaction. So it is called as lock and key method. So substrate is the key, the active site of the enzyme is the lock. So this theory explains the specificity of enzyme, that how specific enzymes are. This explains the loss of enzyme activity when enzyme denature. So when enzymes are denatured, they will not do any kind of activity. When enzymes are denatured, they will not do any kind of activity. It is very essential and that enzymes should remain in their active state. Okay. Then we study two terms, apoenzyme and holoenzyme. What is the apoenzyme? Enzyme without its non-protein part. What is a part of enzyme which do not have its protein part is called as apoenzyme. And what is holoenzyme? Holoenzyme is an active enzyme with its protein content. So when another molecule called as coenzyme, this coenzyme is called as cofactor because it is the activated part of apoenzyme. Activated part of apoenzyme is called as coenzyme, and when this apoenzyme plus coenzyme mix, they will form holoenzyme, that is called as whole enzyme, which is this protein part. So active enzyme side, which is non-protein part, is called as holoenzyme. So what is apoenzyme? Non-protein part. What is coenzyme? Activated part of non-protein part of enzyme. And when both together meet, they will form holoenzyme. So apoenzyme plus coenzyme is equal to holoenzyme. All are having non-protein part mainly. So the role of cofactor is that it is important for part in the carrying out of chemical reaction. Like vitamins are very good example of cofactors. They are this in certain biochemical reaction. Okay, then we define what is cofactor. So the cofactor is a non-protein chemical compound which is tightly bound or loosely bound to the enzyme which is required for catalysis. Cofactor is a non-protein chemical compound which is either tightly bound or either loosely bound towards the enzyme for its activity. Based on their bounding, there are two types of cofactors: coenzyme and prosthetic groups. Coenzyme and prosthetic groups. So there are two types of cofactors, coenzymes and prosthetic groups. Okay, then we discuss what are coenzymes. So that non-protein part which is loosely bound to apoenzyme. The non-protein part which is very lightly or loosely bound to apoenzyme by non-covalent bond. By non-covalent bond is called as coenzyme. Like vitamins or compound derived from vitamins. Like vitamins or compound derived from vitamins. The non-protein component which are loosely bound to apoenzyme are called as coenzymes. Main example is vitamins or compound derived from vitamins. Then next type is prosthetic group. It is again the non-protein part, but it is tightly bound to apoenzyme by covalent bonds. Because when covalent bond is there, definitely it will bound tightly, and when this non-covalent bond is there, it will bound very loosely. So that non-protein part, which is tightly bound to the apoenzyme by covalent bonds, are called as prosthetic group. What they are called as? They are called as prosthetic group. Okay. Then we discuss about enzyme specificity. All enzymes having different degrees of substrate. They may recognize single substrate, group of substrate, or particular type of bond. Because they are substrate specific, they can recognize single substrate, group of similar substrate, or particular type of bond. Okay. Then we discuss certain types of enzyme, their reaction time and example. First type is absolute. Absolute enzyme, they can catalyze only one type of reaction for a single substrate. So absolute enzymes, they can catalyze only one type of reaction for the single substrate. For example, urease. Urease only will catalyze the hydrolysis of urea, not any other chemical. Second type is group. It can catalyze one type of reaction for similar substrate. They can catalyze one type of reaction for similar substrate. Example is hexokinase adds to phosphate group to hexoses. Example is addition of phosphate group to hexose by the hexokinase. Third is linkage. Linkage catalyze one type of reaction for a specific type of bond. Linkage catalyze one type of reaction for a specific type of bond. Example, chymotrypsin catalyze the hydrolysis of peptide bond. Okay, chymotrypsin catalyze the hydrolysis of peptide bonds. Right? So what is the example? Chymotrypsin which catalyze the hydrolysis of peptide bonds. Then what is activation energy? We discussed that enzymes lowers activation energy. So all chemical reactions require certain amount of energy to get them started. This energy is called as activation energy. It is the first push to start the reaction. Suppose we push the door to get outside. So some chemical reactions require certain energy to start. They require certain push. So that push means that energy which is required as a push is called as activation energy. So that energy which required as a push is called as activation energy. So enzyme lowers down the activation energy when they initiate the biochemical reaction. Now see the structure of enzymes. Very simple. In a simple flowchart we can explain. See, this is the enzyme. Simply part is only protein. Complex part contains holoenzyme, which is proteinous part, and non-protein part, that is cofactor. Complex part or holoenzyme, it means they contain both protein and non-protein part. They act as cofactor. And the protein part of this non-protein part is called apoenzyme. So this protein part is called as apoenzyme and non-protein part is called as cofactor. Now this cofactor, I told you two types, coenzymes and protostic group. Coenzymes were loosely bound to the apoenzyme. The example is large organic molecules. And in prostatic groups, what we discuss, they are tightly bound apoenzymes. They contain small inorganic molecular atom because it is difficult to control large molecules. So they are not tightly packed. So they are only loosely bound. For small molecules, suppose we want to put small item in a bag, we can easily put that. But if you want to put big item in the bag, it is not possible. Same concept applies here. If the molecule is small, it can be easily tightly bound to the apoenzyme. If molecules are big, they are loosely bound to apoenzyme. That large molecules are called as coenzymes. Small inorganic molecules or atom are called as prostatic groups. One thing important, large molecules are organic, small molecules are inorganic, means they contain some metallic ions. Well, large organic molecules made up of carbon, hydrogen, oxygen. So, there are two types of cofactors, that is prostatic group and coenzyme. Very simple diagram is there, we can easily understand. Then we discuss about the mechanism of action of enzymes. As I told you, enzymes increase rate of reaction by the decreasing of activation energy. Okay, so both are inversely proportional to each other. And I told you enzyme is reacted by enzyme substrate reaction. Lock and key method we have discussed. Induced fit method we have discussed. In lock and key method, substrate is key, active site is lock. So substrate has to mesh the size of the active site so that it can penetrate into the enzyme and it can perform the reaction. What is called as lock and key method. And there's an induced fit method means substrate actually fit inside the active site of the enzyme. Okay, then we'll discuss about induced fit model. What is that? In the induced fit model, the active site is flexible, it is not rigid. This is the enzyme, this is the substrate, this is the active site, which is open. The shape of the enzyme active site and the substrate adjust to maximize the fit. So when substrate is added, it will automatically shape its conformation so that it can fit inside the enzyme. So it is called induced fit model. And finally, they will form what? Enzyme substrate complex. So the shape of the enzyme, active site, and the substrate adjust to maximize the fit. They will improve catalysis of the enzyme. There is greater range of substrate specificity. There is greater range of substrate specificity. This model is more consistent with the wide range of enzymes. This model is more consistent with the wide range of enzymes. Okay, so these two theories are what? Small fit method, induced fit method, Hawkins method. Okay, these are two main concept mechanisms from which enzyme reacts. Then, now we study how does this enzyme catalyst reaction takes place. Suppose there is substrate S. It fits in the active site of the enzyme. So what it will form? It will form enzyme substrate complex, means E plus S is equal to ES. Then within the active site, when this complex has the active site, ES will again break into E plus P. So this enzyme substrate complex will convert this substrate into the product. So then they will form E plus P. E plus S is equal to ES, ES is equal to E plus P. So substrate is converted into the product. The products are then released, allowing another substrate molecule to bind into the enzyme. And this cycle means E plus S, ES, ES into E plus P. This cycle repeated millions of times, blah, 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 blah. So, what is the oral reaction? Oral reaction is that E plus S, they will form ES complex, and they will convert into E plus P. So finally, the product is completed. We can see this diagram. Enzyme plus substrate, they will form the complex. This is the substrate. It is fused inside the enzyme. It will form what? Enzyme substrate complex. It is the first step. In the second step, ES is converted into the EP. So enzyme substrate is converted into the enzyme product. So enzyme substrate, it goes to the transition state, and then finally it will cause to the productive stage. ES goes under the transition state, and then from the ES it will form the EP, that is productive stage. They are called as enzyme product complex. Then EP finally form E plus P. Enzyme is ready for another once the product is out. After that, what will happen? Another substrate will come. Again, they will form E plus S. They will form ES, and then from S again they will convert into the product. So this cycle keeps on changing. Okay, so this cycle keeps on changing. Then, how the enzyme activity is affected? I will study which are the factors which affect enzyme activity. First are environmental conditions. Second are certain cofactors and coenzymes. Third are enzyme inhibitors. We will discuss each in detail. First will be what? Environmental conditions. Second are cofactors and coenzymes. And third are enzyme inhibitors. So these factors will affect the enzyme activity. Okay. Now first we will discuss about the environmental conditions. As we know, they include temperature, pressure, pH, moisture, humidity. So in that, first we will discuss about temperature. So extreme temperature, whether too low or too high, they can be problematic for enzymes. So extreme temperature are the most dangerous. They can denature the activity of enzymes. So as a result, what will happen? Enzyme will not activate the substrate and it will not convert it into the product, and the reaction will not be completed. Second is pH. Enzymes normally are active in the pH range of 6 to 8, means neither too acidic nor too alkaline. Change in the pH, enzyme will deactivate. And then upon the substrate concentration, more substrate, more product, less substrate, less product. So these three include the environmental condition. Then we discuss about cofactors and coenzymes. So inorganic substance like zinc and iron and vitamins, they are sometimes needed for proper enzymatic activity. If there are no cofactors and no coenzymes, enzyme activity will not perform. So we can say that enzymes directly dependent on cofactors and coenzymes. Example is iron must be present in the quaternary structure in hemoglobin to pick for oxygen. We all know that our blood contains hemoglobin. It absorbs oxygen. Then there is a transport of blood in the lungs. And all it is based on enzyme activity. If the hemoglobin do not contain the iron, what will happen? The color will not be red. And if hemoglobin is decreased, blood circulation is affected in the lungs, in the heart. It can cause death also. So cofactors and coenzymes are very, very important. Adequate supply of cofactors and coenzymes will increase the activity of enzymes. So their activity is very, very important. So what I've quoted the example, iron must be present in the quaternary structure in hemoglobin in order to pick the oxygen. Then we discuss about substrate concentration and reaction rate. So the rate of reaction increase with the substrate concentration. We can see this diagram. This is the substrate concentration. This is the initial reaction. See, lot of substrates are coming. Enzyme is saturated. Maximum activity occurs when the enzyme is saturated. When all the enzymes are binding saturated substrate, they will continue their reaction. But after some time, activity will be inhibited. But enzyme activity increasing, all the enzymes are included in binding with the substrate. It keeps on increasing, and later on it becomes saturated. So substrate concentration is directly dependent on the enzyme. Enzyme activity will increase with the concentration of with the increase in the concentration of substrate, because more substrate, more product. But after some time, it will show some saturation. Okay, everything has some growing phase and declining phase. So after some time, it will show some saturation. So this diagram clearly shows the amount of substrate, the velocity of enzyme, it is the maximum activity of enzyme, increasing order of the enzyme substrate activity. Okay, so then we discuss about what are the enzymes inhibitor, how the certain inhibitors can stop the activity of enzymes. So first, we will discuss about competitive inhibitor. Competitive inhibitor. This inhibitor is same structure like substrate. It will fight with the substrate. So when it will fight with the substrate, if inhibitor wins the battle, it will directly go attached to the enzyme. As a result, what will happen? The activity of the enzyme will decrease. So has its effect reversed by increasing the substrate concentration. So I will explain one again. Suppose there are two enzymes. The shape and size of inhibitor is exactly the same in size of substrate. So both will fight with each other. If the inhibitor wins the battle, it will reach to the surface of enzyme. It will deactivate the enzyme, block the enzyme. But how can we overcome this problem? We will increase the substrate concentration. So what will happen? The increasing population of substrate will kick away the inhibitor from the enzyme. What will happen again? The enzymes can become activated. The competitive inhibition can be controlled by increasing the substrate concentration. So what I told, that competitive inhibitor can be controlled by increasing the substrate concentration. Then we discuss about non-competitive inhibitor. It has a structure different from the substrate. So this non-competitive inhibitor, it has different structure from that of substrate. So it will not fight with the substrate. What it will do, it will directly come on the enzyme. It will disturb the shape of the enzyme. It will alters the active site of the enzyme. It will cover the active site of the enzyme. It will not allow any other substrate molecule to bind on the enzyme. So it is the permanent inhibition. We cannot reverse a non-competitive inhibition by increasing the substrate. So here what is the opening? There is an inhibitor. Structure is different from the substrate. It will not fight with the substrate. It will directly come on the active site of the enzyme. It will change the structure of the enzyme. So once it will change the structure of the enzyme, it will prevent the binding of the substrate on its active site. Why? Because the active site is already covered by the inhibitor. So no matter whether you increase a lot of substrates, they will not able to attack on the inhibitor because it is already occupied the active site. So this type of inhibition cannot be reversed with increasing substrate concentration. Okay. Then we discuss about certain nomenclature of enzyme. Every enzyme is ending with ASE. Oxidase, catalase, reductase, all enzyme ending with ASE. If this enzyme called sucrase, it will catalyze the hydrolysis of sucrose. If it will catalyze the sucrase, it will catalyze the hydrolysis of sucrose. So the name describes the function of enzyme. Oxidase, it will do oxidation reaction. Sucrase will do hydrolysis of sucrose. Lactase, it will do hydrolysis of lactose. So there are some common names which are not based on a chemical reaction of digestion of enzymes, like pepsin and trypsin. Okay, so some common names which are used particularly for the digestion enzymes, they are called as pepsin and trypsin. Now these names are different from the chemical reactions which they are doing. Like pepsin and trypsin basically attacks in the intestine for the digestion of food. Okay, for example, some enzymes describe both substrate and the function, like alcohol dehydrogenase. So what it will do, it will attack on which substrate? It will attack on alcohol. And what it will do, it will cause dehydrogenase. It will cause the oxidation of dehydrogenase. So what is the name of enzyme? Alcohol dehydrogenase. So certain enzymes can describe both the substrate and the type of reaction. So we know that in this enzyme, it is acting the dehydrogenation of alcohol. So the name suggests alcohol dehydrogenase. So every enzyme is ending with ASE, and by their name we can identify the reaction which they are catalyzing. Like lactase, they will cause the hydrolysis of lactose. Sucrose, they will catalyze the hydrolysis of sucrose. Oxidase, they will catalyze the oxidation reaction. Then International Biochemical Committee. Okay, enzymes are classified into 6 functional classes based by the International Union of Biochemists, that is IUB. Okay, this International Union of Biochemists, they have showed that enzymes can be categorized only in these 6 classes. There are no any other classes besides these 6 classes. Like EC1, it is called as oxidoreductase. What it will do? It will do both oxidation as well as reduction. It will call as EC1, Enzyme Classification 1. EC2, its name is transferase. As the name suggests, it will transfer one functional group from one metabolite to another or one molecule to another. Third is EC3, hydrolysis. It means what? It will do the hydrolysis of water, split the hydrogen ion, oxygen ion. Okay, EC4, it is called lysis. Lysis means it will do the cleavage or it will digest certain molecules. Okay, lysis means to digest something, to cleave something. Okay, fifth group is EC5, that is isomerase. What are isomers? Which are structurally different and functionally similar. So it will cause the structural change in the same class of compounds. EC6, it is called ligase. Ligase means to join something. So this International Union of Biochemists, based on these 6 basic chemical reactions, they have classified the enzymes. Besides this, we don't think there are any other activity of enzymes, right? If suppose we talk about EC4, which activity is carried by EC4? We will call as lysis. It is digestion of certain compounds. Okay, so there are total 6 type of this classification of enzymes. Then next comes principle of the classification of enzymes. Okay, principle how we can further classify the enzyme number. Suppose first example is EC 2.7.1.1. Suppose first example is EC 2.7.1.1. This enzyme denotes hexokinase. This enzyme denotes hexokinase. Now, why it is called 2.7.1? Because these components indicate the following group of enzymes. 2 means second group was EC2, that was transferase. Okay, 7 is subclass. Subclass is transfer of phosphate. The EC2 means it belongs to second class. First was oxidative reductase, second was transferase. 7 is subclass, that is transfer of phosphate. 2.7 is clear. 1 is subclass because alcohol is the acceptor of this phosphate. Alcohol is the acceptor of this phosphate. So 2.7.1. Last point 1, it is a specific name. So what is the name? ATP dehexose 6-phosphotransferase. So it is called as hexokinase. ATP means it is doing a transfer of phosphate. What is subclass? Alcohol phosphate, and specific name 2.7.1.1. So it is called as ATP dehexose 6-phosphotransferase, which is called as hexokinase. Okay. Now, what is the importance of hexokinase? See, it contains magnesium ionized catalyst as cofactor. It converts ATP into ADP. So this hexokinase will do what? It will convert glucose into glucose 6-phosphate. Means glucose is transferred into glucose 6-phosphate with expenditure of certain energy. ATP, adenine triphosphate, is converted into ADP, and phosphate group is attached to the glucose molecule. So this is the isomer of glucose, and it has attached the phosphate group. So hexokinase will do the catalysis of glucose to glucose 6-phosphate with the expenditure of some energy. Then another class, oxidative reductase. It will catalyze what? Oxidation reduction reactions. Typical subclass, oxidase reductase dehydrogenase. Function, oxidation, reduction, and in dehydrogenation, what it will do? It will remove 2 hydrogen ion to form double bonds. Example is when ethanol reacts with coenzyme in the presence of alcohol dehydrogenase, it will form acetaldehyde. So ethanol plus NAD, it will form acetaldehyde. It will form NADH plus H plus. So both hydrogen ion are transferred to here. Then come transferase, transfer of function groups. Transaminase kinase, it transfer what? Amino groups and phosphate groups. So alanine plus alpha ketoglutarate. In the presence of alanine transferase, they will form pyruvate plus glutamate. So here there is transfer of amino group and there is transfer of phosphate group. Next is hydrolysis. It will do the hydrolysis of water. Example, peptidoglycan, lipase, amylase. They will hydrolyze peptide bond, hydrolyze ester bond, or hydrolyze 1,4 glycosidic bond. Their main function is to hydrolyze the bonds, that is peptide bond, ester bond, or hydrolytic bond. So here polypeptide bond is there. Hydrolysis comes into the play. You will form short polypeptide, and it will release the amino acid from the C terminal. Then say lysase, addition of group of double bond or removal of group. Which subclass? Decarboxylases, dehydrase, deaminase. It will remove CO2. Dehydrase will remove H2O. Deaminase will remove NH3. So pyruvate is converted into the acetaldehyde plus CO2. in the presence of pyruvate decarboxylase. So pyruvate in the presence of estrous ion, in the presence of enzyme pyruvate decarboxylase, it is converted into acetaldehyde plus CO2. Next is isomerase. It will do what? Just only the rearrangement. Examples of isomerase and epimerase. They will convert into cis form and trans form. They will change into D form and L form. D I told you that rotatory, L is lever rotatory. Right hand side direction, left hand side direction. They will change the position. So here, malate is changed into fumarate. Suppose this malate CEOs comes here, S comes there. So it comes malate to fumarate. It is the example of isomerase. Then come ligase to join something. It will do bonding of molecule using ATP energy. Suppose pyruvate with the help of CO2, with the help of ATP, in the presence of pyruvate carboxylase, they will form oxaloacetate plus ADP plus H plus ion. So this is the example of ligase. So all enzymes can be identified based on the reactions they perform. Now last is mathematical identification to study how to study the mechanism of enzyme mathematically. So there is a scientist called as Michel Menton. It gave the model to study the basic chemical kinetics of the enzyme. Michel Menton kinetics is one of the most simplest and best known models of the enzyme kinetics. This model serves to explain how an enzyme can cause kinetic rate enhancement of the reaction and why the rate of reaction depends on the concentration of the enzyme present. Why the rate of reaction depends on the concentration of the enzyme present. Okay? It depends upon why the rate of reaction depends on the concentration of the enzyme present. So to begin our discussion of enzyme, let us define the number of moles of products P, which is formed per unit time as V. So it is the variable is V. The V is also called as the rate of catalysis of the enzyme. We can say V as a variable, which is the rate of catalysis of the enzyme. So for different enzymes, obviously the V varies with the different concentration of the substrate. So at low substrate concentration, this variable is linearly proportional to the substrate. But when the substrate concentration is high, then it is not effective. Then V is independent of the substrate. It can work up to certain limit only. I told you in the case of competitive inhibitor, how we can overcome that by increasing the substrate concentration. But when the substrate concentration is low, variable is directly proportional to the substrate. But when the substrate concentration is high, then it is V is independent of the substrate. Then see, we will use general enzyme chemical reaction, which includes back reactions in addition to the forward reaction. Enzyme plus substrate. K1 means forward backward reaction. They will form what? Enzyme substrate. This reaction could be forward and backward, so double arrows are there. And finally, this ESC will go in the forward direction. It is having constant of K2, and this it will convert into the enzyme plus product. Thus, it will convert into the enzyme plus product. So to understand Michel-Menton kinetics, we will use the general enzyme reaction shown below. So enzyme, when reacted with substrate, they will form enzyme substrate. So they can also be reversed again to form E plus S. So that's why it is the reversible reaction. That's why it is a reversible reaction. And when E plus S goes to E plus P in the form of K2, they will form enzyme plus product, enzyme product. They will form what? E plus 2, that is enzyme plus product, E plus product. So this reaction only goes in the forward direction. E plus P cannot form ES, but ES can form E plus S. So this is the forward reaction. Then see some substrate complex. E plus S is equal to ES, V is equal to 0. It is directly proportional to the K1 ES. ES back into E plus S. Again V0 is K2 directly proportional to the ES. ES converted to the product K3. Here V0 is directly proportional to the K3 in the ES. E plus P K4 back reaction ES V is equal to K4 EP is equal to 0. So the ES complex is formed by combining enzyme with the substrate at the rate constant K1. The ES complex can either dissociate to form EF and K2 and K3 respectively. In the case of rate constant K4, enzyme plus product goes into the forward direction. Again, they are converted into the substrate. So here, the velocity will be 0 because this reaction is not favored. So this lecture, we completed about the enzyme. So in this lecture, we'll understand better about the enzyme, types of enzyme, their classification, their basic nature, their mode of action, their chemical reaction, factors affecting the enzyme, and mathematically to understand how the enzyme kinetics work. So thank you very much.